Welcome, everybody, to Fungi and Trees, or What's New in the Hidden Forest, Looking at Trees with Susan Goldhor. It's a joint program of Cook Memorial Library and Chocorua Lake Conservancy. I am delighted to introduce Susan Goldhor, many of you, I feel like I should start the recording again. What do y'all think? Who many of you have the good fortune to know. Susan is a biologist who got interested in fungi and plants after she retired from working with animals. The Boston Mycological Club provided her introduction to fungi. In return, she served as president for 12 years. Her mycological essays have been widely published, and she's a contributing editor at Fungi magazine. Her major interest has always been fungi in our forest ecosystem, but as anyone who spends time with her can attest, her interests are wide-ranging, and one of her many strengths is connecting myriad diverse dots. I feel tempted to make allusions to mycelia, but we're so lucky to have her here with us tonight. Thank you very much, Susan, for being with us.
Wow. Thank you, Juno, for that great introduction. And thanks to the Conservancy and also for the Tamworth Library and Mary Cronin, who's my technical advisor on all things Zoom. So I'm going to start out by introducing you to just some terms that I want you to know. These are the lifestyles of various fungi in their relationship to trees. So a saprobic fungus is one that rots dead trees or dead wood. Pathogenic fungi attack and kill trees. Mycorrhizal fungi are mutualistic with the roots of living trees. And endophytic fungi are those that live in the organs of trees without causing disease. So just to show you some visuals of examples that you might see in our forest, here's a saprobic, a lot of saprobic fungi. Here are some pathogenic fungi that you're probably familiar with. Some mycorrhizal fungi. And in all of those cases, in the rare events when you see a charismatic mega fungus, the real fungus is the mycelium that they grew out of, and each individual thread in this mycelium is called a hypha. And as for the endophytic fungi, you won't see them because they're microscopic and mysterious. So my idea is that we're going to look at the tree in terms of its three major sections. There's the canopy, there's the trunk, and there are the roots. And we're going to go top to bottom and talk about each section. And I've been cutting down and abbreviating this talk, and I've tried to pick just some aspects of each section that I think are interesting and important, and where we have some new information over the last few years that you might not know. So, starting from the top, we're going to talk about endophytes. And endo means inside, and phyte is plant. And these are microscopic organisms that live within the leaves or other organs of plants without causing disease. And we're really, in a way, talking about the plant's microbiome. In our case, our microbiome is pretty much all bacterial. In the plant's case, it's largely fungal to the extent that it's sometimes called a mycobiome. So endophytes are everywhere on the tree. I mean, literally everywhere. No matter where you are, whether you're in a field of crop plants or in a forest, every leaf, every petiole, every twig on every single plant, everywhere in the world, except of course Antarctica, is full of endophytes. And that's another thing that's interesting, is that if you looked at a graphic of our microbiome, it would really just be on the surface. You would see like an outline of us, plus the organ systems we have, like our GI tract, that are connected with the outside. But in the case of a tree, you might see a graphic of the microbiome of a tree as pretty much a solid. So endophytes were originally discovered about 150 years ago, and they were discovered in largely domesticated plants, and they were obvious because these were microorganisms that were in every cell of the plant, including its germplasm that made seeds, so that they were carried from generation to generation. And they conferred unique traits upon these plants. So, for example, sleepy grass was something that produced a toxic compound that literally put an herbivore to sleep for like a day if it ate it. And I would say a sleeping herbivore is usually a dead herbivore. They enabled tomatoes to grow at 140 degrees. They enabled plants to do things like survive in drought or survive in very polluted environments, deal with salt. They got people very excited because immediately people saw ways to make money out of these things and to use them for human benefit. But these endophytes turn out to be unique in both the traits and the fact that they're carried from one generation to the next. Most, and probably all, of the endophytes in leaves in our forest seem to be random drifters from the environment, and they change every season. So when the new foliage comes out in the spring, it has no endophytes. The leaves are new. The endophyte richness and the population builds up over the growing season. And then, of course, in winter, the leaves drop. So if you wanted to look at the endophytes in a leaf, and I should say right here that a lot of the work that I'll talk about endophytes comes out of the research and the lab of Betsy Arnold in Tucson. And she has done a tremendous amount of important work on this subject. So if you wanted to look at the richness of an endophyte population on a leaf, it would actually make sense to go to the tropics, where you might, first of all, find very large leaves, and you might also find leaves that had a very long growing season. So here's an experiment that came out of Betsy's lab. All of those little patchwork pieces that you see are pieces that have been cut from a single leaf. So there are 200 tiny samples of this leaf, and there are 15 samples from the petiole. Anything that's blank was a piece where they couldn't identify a fungus, which does not mean that there were no fungi on it. It may just mean that they couldn't decide, they couldn't see it. It didn't grow in time in the culture. They couldn't identify it. But they basically found over 20 fungi on this single leaf. Now, they also, they actually looked at a number of leaves. This is simply a typical one. And no two leaves were exactly alike, although they were similar. The patterns of fungal growth are random. There, for example, it's not as if you see particular species in particular parts of the leaf. It's really seems that the endophytes, the fungal spores, are simply landing on the leaf, and wherever they happen to land, that's where they're going to make their home. So how do they get in? Well, there's the polite way to enter, which is through the natural stomatal openings of the leaf surface. And this is a pretty amazing micrograph that shows a spore with a hypha growing out of it, and the hypha is growing into the interior of the leaf. But not all spores are polite, because endophytes often include pathogens. And if the pathogen wants, it can break in. It has something called an apressorium, which is basically its drill, and it can just enter the cell. Notice the melanin layer. Fungi use melanin as protective and hardening. By hardening this little pressure organ, they can build up more pressure in it. So this is just a quote from a book on endophytes. And it's interesting that it's hard to imagine general rules, because there are so many phylogenetically diverse endophytic fungi in the foliage of any single host. So here's a question. We've always kind of assumed that the endophytes provide protection against pathogens. Do they? Well, again, we're going to go to an experiment that Betsy Arnold did. This is cocoa. It's a cocoa plantation, and people get funded to work on cocoa because cocoa is economically important. It also is constantly under attack by fungal pathogens, serious pathogens. So there's a lot of interest. So what Betsy did is she looked at cocoa tree leaves as they first came out and then as they developed. When they first came out, despite the fact that they had no endophytes within them, they did have defense chemicals in them that would protect them against pathogens. As the endophytes built up over the growing season of the leaf stopped producing defense chemicals. So the takeaway from that is that the endophytes are protecting against pathogens, that the tree could produce its own defense chemicals, but as anyone who's ever looked at our nation's budget knows, defense is extremely expensive. Maintaining a standing defensive armament is extremely costly. And the leaf is not doing that. It's only doing that when it absolutely has to, when it's young and tender, it has no endophytes. At this point, it needs to expend the energy to do that. But as soon as it gets endophytes, it stops producing its own defense chemicals. Would that we could do the same. But how, if the endophytes are protecting the leaf, how are they doing that? And especially since they're, as far as we know, they are just random drifters from the environment, and some of them actually are pathogens. There are 3 theories about how they do it. One is that some of the endophytes are themselves manufacturing defensive chemicals. Another is they're activating the plant's own defenses, but given that the leaf has stopped producing defensive chemicals, this seems a little unlikely in this case. And the third, which I have to admit is my favorite for non-scientific reasons, is that they are just taking up so much space that there's no room for the pathogens to attack. So, you know, in all this 150 years of our knowing about endophytes, the question asked has always been, what is the plant getting out of the endophyte? What are the endophytes doing for their host? But recently, a small number of researchers have started to ask, what's in it for the endophytes? Because it doesn't really seem as if the endophytes are getting that much out of it. I mean, they're getting a little bit of sugar, not really very much. And then autumn comes and the leaves wither and drop to the ground. So from the viewpoint of the endophyte, it's not necessarily great. But the new theory, which has 2 names, it's called the biophyte hypothesis, and it's also called the foraging ascomycete hypothesis. what these theories propose is that saprotrophic fungi, that is, fungi that can digest wood, and that's how they make their living, can utilize leaves both for dispersal and as a resource haven during dry seasons that don't have a lot of woody debris. And these are very interesting, and it really makes some sense. One of the big problems that fungi have is dispersal. Their mushrooms, for example, over evolutionary time have invented amazing mechanisms for getting their spores out there. And yet, in the forest floor, where there's very little wind or air current movement, if a spore can land 18 inches from a mushroom, it's doing really brilliantly. Imagine if that spore is on a leaf. I mean, all of a sudden, 18 feet is nothing. I mean, the leaf, if you think about it, is a great vehicle for dispersal of spores. The idea behind the resource haven is that summer tends to be a dry month, a dry season, and there are fewer storms that are bringing woody debris down to the forest floor. The idea is that in autumn, when the leaves fall, a lot of woody debris is also falling. It's a moister season, and it's a good time for a saprophyte to be on the forest floor. So basically, I just want to go back. Basically, these theories say that it's this that a lot of the endophytes are there for. And they're not really there to help out the tree. The tree for these endophytes is a summer home. It's just a part-time place to take refuge. Before we leave the canopy, I just wanted to mention that it won't surprise you to know that the leaf surface, which is called the phyllosphere, is very rich in epiphytes. In other words, the outside of the plant. And to us, the leaf surface looks very smooth, but if you look at it in a micrograph, it isn't smooth at all. It's full of hills and valleys. It's things to hold on to, things to grow into. It's a complex structure. It also is fairly rich in nutrients, because leaves tend to, like roots, tend to exude all kinds of good stuff. What this photograph doesn't show are yeasts, and yeasts are in fact a big part of the phyllosphere's inhabitants. I think it's interesting that yeasts also grow wild on the forest floor, even in the group Saccharomyces, which is where we have our domesticated yeasts. They're found on the forest floor in their real wildlife, but those actually are different yeasts than what we find in the phyllosphere. The yeasts and the other fungi on the leaf surface are also thought to be keeping off pathogens, and it's the same group of theories as we had for the endophytes. They have the additional advantage of consuming any sugars and other things that might attract pathogens. Okay, so back to our question. Let's move down to the trunk of the tree. And I have to say that until recently, I didn't really think of the trunk as having endophytes. So there is a about 10 or 15 years ago, this article appeared, and the title was dramatic enough that a lot of people sort of looked at it. Do all trees carry the seeds of their own destruction? And I would also like to point out the last author listed, Lynn Boddy, is an amazing researcher. A lot of what I'll talk about rot and tree pathogens is her work. I think that she has done more to look at rot in a forest ecosystem than anyone else. And her group, she is based at the University of Cardiff in Wales. Her group has done really interesting work. So in this case, it was a question that they found wood decay fungi latently, so not actively, present in the sapwood of large numbers of trees. And if you're a forester, you know that sapwood is not actually living tissue, but there is a parenchymal layer of living cells there. So 2 years ago, Lynn carried this work further, and what they did was they looked at the sapwood of beech trees and looked for fungal endophytes and the origins of decay. And what they did was they took samples. They used a number of beech trees that were in a variety of geographic locations throughout the UK. And this is how they mapped their sample taking. And the picture on the right shows the guy who's, or gal, I don't know which, who's actually taking the sample. And this is what they found. And it's really kind of surprising. If you look at the list on the right of the types of fungi that they found, it's all over the place. It's completely wild. But what it shows is that, unlike what I always imagined, it's actually not difficult to get from the outside of the tree into the sapwood. And the red and yellow colors show saprotrophs and pathogens So in other words, these are things that can rot the tree. So what does it mean for the tree that it's carrying, to use the phrase they use, is carrying the seeds of its own destruction? What it probably means is heart rot down the line. And heart rot for trees is not as bad as it sounds. The assumption is that these various latent rotters and pathogens are not active now because of the extremely high water activity in the sapwood columns that they're in, and that they're not going to become active until the tree is not in quite such good shape and there's less water activity. So first of all, it doesn't mean that anything's going to happen right away. But the other thing is that even when it starts to rot the tree on the inside, it's not, that rot tends to be very slow growing. I mean, we could be talking like a century between the time that they pick up the pathogens and the time the tree is really experiencing severe heart rot. And heart rot doesn't always kill the tree. In fact, some people feel that, at least under some circumstances, it can actually be helpful. So it's not, an instant death kind of thing at all. So we have trees in our forest that we call snags. And I have to say, this is not a name that I really like. In reading Lynn's publications made me aware that in the UK, they have the concept of the veteran tree. And I really like this, especially the wounded veteran tree. And here's what they say about veteran trees. And this is actually that a veteran tree is not always a tree that's really, really old. It's a wounded tree. It's a tree that has been traumatized in various ways, although it may also be of just a very, very old tree. So things like rot holes, dead wood, split trunks, places where major branches have fallen off, and that veteran trees have a different definition and a different use in the forest to just an old tree. That veteran trees always have decay. And it's interesting to note, they say wood decay is a natural process and not usually a sign of poor health. Decay actually increases the wildlife value of veteran trees. So for both food and shelter, the veteran tree, the tree with holes and ragged edges and decay of various sorts in various places, is a very valuable tree to the forest ecosystem. So again, what Lynn decided was important for conservationists is to develop the ability to make veteran trees. And the idea behind this was to have two types of conservation. One was to conserve rot fungi in areas where they were rare or totally absent. And the other was to, and this is a new term that I learned, to veteranize trees, i.e., to make them sort of artificially partly rotten, to wound them, to they are, you can imagine that there are scenarios where you have forests that are lacking in diseased, decaying trees. I have to say, our forest has lots of problems, but one thing that it does have is rotten trees and lots of rot fungi. I'm happy to say that. But imagine a clear-cut area where there might be a few trees left standing, or especially a place where an artificial plantation has been planted and everything is more or less the same age. This is actually a really dangerous and unhealthy setup for a forest. So the highlights of this paper was that to develop methods to both veteranize trees, to create habitat for various animals, and to translocate and in fungi that are rare or threatened in particular environments. So the trunk on the left is having a heart rot fungus translocated into it. It's, in other words, giving it something it may not have. This looks like it's just wooden plugs, but they are plugs that have been carefully inoculated with the fungus in question. And then B shows they're veteranizing a beech with a white rot fungus, and they have they've made a cut in the trunk with the chainsaw, and they're actually stuffing the fungus and sawdust into it. And C is they're translocating and veteranizing with multiple insertions of a rare hericium heart rotter. This is a fungus you might know as lion's mane, but it's a rare species of it. So these are rot fungi that fruit and I'm sure that you've seen all of these in our forest. But they're really a minority. There are thousands of invisible rot fungi. A project in Norway to count the species in decaying Norway spruce logs found up to 400 species on a single log. At the beginning of the decay, these were mostly species that they recognized, but as decay advanced, they found more and more unknown types of fungi. Why are there so many? I mean, it does take a village to rot a tree, but there are a lot of problems with digesting a log. And there sort of are three major problems. One is that a fallen tree has a very high, it has a very low surface to volume ratio. So how do you get in? Of course, towards the end, it's a lot easier for multiple species to get in. Another is that it's just difficult to digest. And the third reason is that it's very, very poor in nitrogen. So, why is wood so hard to digest? It's not cellulose. Wood is lignocellulose. Cellulose is easy to digest. Even about a quarter of humans digest cellulose, so this is not widely known. It's just if you have a slightly longer than normal digestive tract, which gives time for the cellulase enzymes to do their work and to develop. But cellulose in wood is sort of trapped in a cage of lignin, and lignin is really hard to digest. Lignin, it's almost like a plastic. It's been compared to bakelite. Bakelite was the early plastic that your grandparents' black telephones were made out of. And you know how amazingly resistant to breakdown that is. So, in fact, lignin is so difficult to digest that there is no such thing as a lignase or a ligninase. There's no enzyme that breaks down lignin. The fungal and bacterial enzymes that break it down are peroxidases. They basically create peroxide, which, as anyone who ever has bleached their hair or anything else knows, is a really powerful oxidizing agent. So they are basically kind of exploding the lignin apart. We talk about brown rot and white rot. And you see, when you walk around our woods, you'll see examples of both of these. So a brown rotter digests the white cellulose and leaves the brown lignin. This is typical of conifers. They are better at digesting cellulose than lignin. And white rotters, which are more in deciduous trees, digest the lignin and leave the white cellulose. Notice the blue arrow. It's pointing to a circle of melanin. Woodworkers call this spalting, but Lynn Body was the person who who pointed out that these are white rot war zones, that every single area in this wood, which is demarcated by a black line of melanin, is the turf of a white rot fungus, which is saying, this is mine, I'm going to eat it, I'm not going to let you in. when woodworkers basically arrest spalting in an intermediate stage, if it goes to the end rot, to the end stage, this is what it's more likely to look like. It's really pretty much pure cellulose. So the other real difficulty about fungi or any organism trying to make a living out of wood is that whether you're looking at the cellulose or whether you're looking at the lignin, you are looking at carbon, hydrogen, and oxygen. There is no nitrogen. So imagine that you're a fungus and you're trying to make a living in wood. I mean, obviously, you're not going to just attack a, a plank or something like that. You're going to have to wait for decay to start. Because wood itself, if you've ever composted, you want a carbon to nitrogen ratio of, say, 20. That would be ideal, but 50 would work. But in wood, the ratio is between 200 and 500. So it doesn't work. So, and fungi need a lot of nitrogen. They not only have to make protein as we do, but they also have to make chitin. Now, chitin is the structural chemical of fungi. This is what gives them structural strength, and they need a lot of it. They're not the only organisms that make chitin. I mean, chitin is the exoskeleton of insects. It's the exoskeleton of things like shrimp and crabs and lobsters. And cellulose is the most abundant organic material produced on Earth. Chitin and lignin are competing with each other for second most abundant organic chemical. So they need nitrogen. And how do they get it? They can attack and digest another fungus. They can attack and digest nitrogen-fixing bacteria. They actually can incorporate live nitrogen-fixing bacteria into their bodies. Of course, by the way, different fungi are following each of these methods. No fungus is doing all of them. Fungi can do something which humans only do when they're close to death, and we don't survive it, which is that fungi can break down a part of their bodies and then translocate that material to their growing edge. But an easy way to do it is to become carnivorous. And we're learning more and more about carnivorous fungi. This is my favorite carnivorous fungi are the oysters. The oysters are a huge family, and they are not only the Ostriatus, the Pleurotus family, but also Hohenbuehlia, which is a related family. And both of these big families of many species, for all of them, killing and eating nematodes is actually a diagnostic feature. In other words, when you're describing what these mushrooms are like, you'd say tan to white, growing in shelf formation on dead or dying trees, parallel gills, kill and eat nematodes. So the pleurotus, the oyster mushroom that we eat and that we're most likely to find growing here, locates nematodes with specialized hyphae, attacks them, and then penetrates them and digests them. And by the way, fungi don't have sort of bodies which can ingest their food. They're sloppy eaters. They sort of exude the enzyme and then they absorb the digested material. So this is a pretty old photograph. And so what's new? What's new is that last year there was a lot of excitement in certain quarters about our finding out how the oyster mushroom does this. Now here's a more magnified picture of the hyphae of an oyster mushroom, and the white and the black arrows show what are called toxocysts. And these are outgrowths from the hyphae of balloon-like structures which are filled with the venom that they use to attack the nematode, or the nematode probably bumps into it actually, and it spatters it and kills it. I should say that nematodes, in case people don't know, nematodes in this case are a species of tiny worm. They are everywhere where there's dampness and rot. If you pick up a handful of damp soil, you're picking up hundreds, maybe thousands of nematodes. Anywhere there is damp rot, including in all kinds of rotten trees, etc., there are tons of nematodes. There are, nematodes form a phylum of their own. There's one species which is killing our beech trees with beech leaf disease. There are nematodes that are parasites. If you've ever had pinworms, that's a nematode. There's a nematode which grows to lengths of more than 30 feet, which is a parasite of blue whales. Nematodes are pretty amazing. It's been estimated that if you could weigh all the nematodes in the world, their weight would be equivalent to about 80% of the weight of all the humans in the world. So don't worry about our running out of nematodes because mushrooms are killing them. So one thing that people were very curious about is what is the venom that the oyster mushrooms that are in the lollipops. By the way, this was how this finding was described in the literature. The carnivorous mushroom P. austriatus has evolved a nerve gas in a lollipop. This is the chemical. It's really kind of a letdown It's nothing. It's this little simple thing. When people found that this was last year, this came out, and people had sort of two reactions to it. One is, this is a very common chemical. It has a pleasant odor. It is used in the fragrance industry. It's made by plants as part, like rosemary and lavender, as part of their scents. It to us, it does not seem at all dangerous. And the other thing is that when you say venom, and people expected a venom, basically, here's a typical venom. I mean, this is a rattlesnake venom. And venoms look like this. They're huge, complicated molecules. It's sort of weird, and people were surprised. However, it does disrupt the nematode surface membrane, and it kills its neurons. So it's doing the job, whether or not we would have thought it was capable. Okay, so moving down, we are now going to look at the roots of the trees. So mycorrhizal fungi are mutualistic fungi that partner with the roots of plants to form mycorrhizas. So mycor means fungus and rhiza means root. And a mycorrhiza is it's like a little growth made by both parties on the root. Here's something that tells you how important mycorrhizas are. That when the first plants came on land, they probably came with mycorrhizas as roots. So there are 3 types of mycorrhizal fungi. People say 4, but I think they're wrong. One is the vesicular arbuscular, which is a terrible name. So I would call those endomycorrhizal. And these are the oldest species. When I said that plants came with mycorrhizas instead of roots, it would be the endomycorrhizal type of fungus that would have been. We think about 95% of the plants on Earth have mycorrhizal partners for their roots, and the endos partner with about 200,000 species of plants, way, by far the majority. The ectomycorrhizal fungi partner with about 6,000 species of plants. And the ericaceous plants, fungi, are really kind of an offshoot of the ectos. They are found only in acidic environments like heath and bog. So they are helping plants like, for example, cranberries or blueberries. The ectos are really the ones that we're interested in. And what's interesting is that we think that there are about 20,000 species of ectomycorrhizal fungi but they partner with only about 6,000 species of plants. But we're interested in them because they are the ones who partner with most trees in our forest. Not all trees on Earth by a long shot, but especially the boreal and temperate zone forests that we have. It is weird that there's so many species of mycorrhizas in our forest. Probably not 20,000, but a lot. We don't really know how many. And we actually don't have very many species of trees in our forest. But what it means is that one tree can have multiple partners. And this shows a tiny section of a tree root hair and with 3 different species of mycorrhiza on it. And you see by the dark and light brown and the white colors, these are all different mycorrhizal species. So the fungal partner is, here's where you get to this thing of trees don't have roots, they have mycorrhizas. The fungal partner sends out these very, very thin hyphae, and these are what explore the environment. They can travel further, they can get into tiny crevices, they can exude the acids or enzymes that are necessary to free up phosphorus from from whatever mineral particle it may be held in. They can ectomycorrhizal fungi often can break down organic litter. They can get nitrogen from the environment. They are really feeding the tree. And the tree, since fungi are genetically and in many ways much closer to animals than they are to plants, and only plants can photosynthesize, the fungi like us are getting their carbohydrates from the trees. So these pictures are pretty much self-explanatory. So the hypha from one tree can join with the hypha of another tree if they are from the same species. And this is a diagram of mycelial connections as some artist has envisioned it. So now imagine a huge web of mycelia joining together. And here we get the famous, now the famous Wood Wide Web. So on the left is a picture of Suzanne Simard, who was the scientist who first did the experiments that showed that the connections between trees formed by fungal webs were passing nutrients between them. And when her article appeared in Science, some editor, some anonymous editor with an incredible PR talent came up with the term the Wood Wide Web. And it comes from that science, that Nature editor. And Suzanne Simard has become like a folk hero. She has become an icon. You know, I don't know how many people have watched her TED Talks. Her experiments were very interesting. She did really kind of two types of experiments. One was where you enclose the canopy of a tree in a gas-tight bag or, plastic thing, and give it radioactive carbon labeled carbon dioxide. And then you can see that radioactive carbon appearing in other trees. And the other type of experiment that has been done, which this sort of shows, is where tree roots were encased in mesh bags that had a mesh that was so narrow that the root couldn't get through it, but the fungal hyphae could get through it. And then material flowed through. So Suzanne discovered socialism in the forest, and it was run by fungi. And the idea was that there was this incredible web, and this is a very, very powerful diagram. The arrow is pointing to the mother tree, by the way. It's actually labeled the mother in the original publication. And people love the idea that the big trees that could access sunlight because they were up in the canopy were feeding sugars via the fungal web to their offspring. And of course, then we found out that that there were all kinds of exchanges in the forest. It didn't have to be the same species of trees, that it was it was great. It was socialism in the forest, with those who had giving to those who needed it. This, by the way, this is a screenshot I took from a TV program, Ted Lasso, where this professional football coach is reading Suzanne Simard's book on finding the mother tree. And in a subsequent program, he quotes her about the importance of socialism and how she showed that it was that's what's working in the forest. What is really amazing about the Wood Wide Web and Suzanne's work is the traction that it has gotten. I honestly don't think that there's any theory that has gotten this kind of public excitement since Darwin came out with Origin of Species. And the difference is that Origin had enemies because the church was against it, whereas Suzanne's theory doesn't seem to have any enemies. I mean, everybody loves it. If you're a socialist, you love the idea that that the capitalism of the sunlight is actually being spread out. if you believe in family values, you love the fact that the young trees are being fed by the mother trees, that it's just, it has something for everybody. So the one thing about this is that I have to say, when I looked at this, I had two thoughts originally. One was, wow, this was a lot of work. If he really took all these samples to work all this out, he earned his PhD. And the other thought was that hyphae are so thin and they're so delicate, and everybody in the soil likes to eat them. I mean, every insect eats hyphae. And there's so many different species of fungi making these hyphae. It was almost too good to be true. Not that I thought that at the time, because I was carried along with everybody else. So uh, my friend Tom Horton sent me, this is the actual data of that plot. And the black dots in here are where he actually, looked at the data. And it's an impressive amount of work. Yeah, that's the mother tree. Yeah, sorry. You can see what it is. But it's not exactly as robust looking as the published diagram. So Tom Horton also sent me his view of the Wood Wide Web. And Tom is he's a mycologist who was one of the very first people to look at the effects of the underground mycorrhizae on how trees move and colonize new spaces in in the forest. And the way Tom sees it is all these different small mycorrhizal mycelial networks, not one simple big network, but a number of tiny networks of different species. So last year, this paper got published, and it was the one of the few scientific publications I know of where the New York Times had a headline about it in its science section before it was even published. And so I contacted Justine Karst, who's the senior author, and talked to her and invited her to speak to our, we have a myco consortium that I'm one of the organizers of, which consists of 20 mycological clubs in this country and Canada. And she was very, very nice and did. Now, Justine is a younger scientist who has a very, very parallel upbringing and career to Suzanne Simard. And in fact, they worked together and have published research together. But she and two of her colleagues, two other young mycologists, read every single paper which talks about, which is said to prove the Wood Wide Web, and which does research which has led to the concept of the Wood Wide Web. And what they found after a huge amount of work, I cannot even imagine going over all these papers the way they did, is not that it's not possibly true, but that it really is not totally proven, that every paper could be explained in a different way, that there are alternative hypotheses and experimental outtakes that could change this view of these massive mycorrhizal networks. So after she gave this talk, which was very long and very intense, when it was over, one guy said to me, he said, "I know, I listened, but I still believe it." So that's where I'm going to leave you. And thank you for staying through this. Now I've got to stop share. Was that going to work?
Thank you, Susan. I know we have at least one question already in the chat, and people are welcome to raise their hands or unmute or add more questions to the chat. Are you ready to take questions?
Yes.
So we have a question. Does chaga kill birch? Does it grow on young trees or at a particular stage of life?
Chaga, something I didn't talk about. I think it generally grows on do not believe anything I say about chaga because I'm not a big expert on it, but I think that it generally grows on older and probably damaged birch.
I had a question when you were speaking about mycelia, bacteria, and yeasts as rich on leaf surfaces. Is this true also of other plants, not just trees, presumably? Kale,
Yes, it's
um,
true of other plants and plants that, there are a lot of plants that have other methods of discouraging pathogens from their leaves. For example, leaves that are hairy, that they're full of little hairs, so that and I would guess that those don't have all those epiphytes.
right. It made me curious. Some people say if you're taking plants from your garden and you eat them without washing them, that this is good for the human microbiome. And I'm curious if part of that is because, we're made up of bacteria and viruses and everything.
Well,
Whether all that mycelia and bacteria are a little extra something for us.
okay, our microbiome is largely bacteria. We have fungi in our microbiome. I'm sorry to say that they're mostly pathogens. The bacteria are holding those fungi down. They're keeping them from attacking us, which is why if you take too much antibiotic, you may get a yeast infection. The yeast has been there all along, but it's been kept sort of inactive by the bacteria. So it's hard to say anything about our microbiome from the microbiome of plants. But but having said that, I don't bother washing a lot of.
And so is it correct that the need for these kinds of heart rot fungi inoculation is because of human activity that's destroyed old growth forest or certain habitats, that if you had forests that had grown for a long time and had all the different ages in them, you'd have veteran trees naturally, and you'd have the fungi naturally. Yeah.
Yes.
Um,
Yes, it would, but I was thinking about things that wipe out a lot of trees, disasters like fires, clear cutting, but often the problem with that is that you don't have any trees to inoculate. You may have lost everything, and you don't have a tree to either inoculate to bring in fungi that you've lost, or a tree to veteranize.
mhm. Well, I really appreciate the way you have shown us yet again the incredible intricacy and interconnection of the other than human world, and the ways that touches the human world. Does anybody have final questions?
Wow.
Then I'm going to say thank you so
Either
much.
everybody got everything or everybody's asleep, one or the other.
I mean, I got some other things. Don't eat wood was one of the messages I took from this.
Well,
Not-
right,
not
don't
I have a question.
actually, yeah, if you're at one point, people were trying to work out a diet for obese dogs. And one thing that was suggested was to add sawdust, because they wouldn't get anything out of it, but it would fill them up. So eating, yeah, but you can eat rotten wood. It's rotten. The point is that rot is what gives us all the nutrients that allow so many animals and fungi and bacteria, etc., to grow on wood. It's all in the rot.
Right, but we should eat the kind that's had the lignin rotted out of it, right? Not the kind that's just had the cellulose rotted out. Okay. So remember that, everybody. Jack, you have a question?
Yeah. I'm thanks, Susan. That was an amazing talk. I really appreciated it. And just this last little exchange between you and Juno was sort of making me think of all the times in this talk that you used kind of very colloquial ways of describing the natural world, like when you said that fungi are sloppy eaters. And I'm just curious if you have used that kind of language because that is what is helpful for lay people to understand what you're talking about, or if you kind of, and people like you, conceptualize these processes in that way.
That's really how I think of it. And I think, by the way, that that probably I think a lot of scientists think of it this way. We're always being told not to anthropomorphize, but I think that anthropomorphizing things is a very natural way for all of us to think about it. And, you know, I guess maybe I could have described that using more technical language if I really worked at it. But I really think of, to me, the natural world is a whole series of stories, fascinating stories about things like rot and competitions and warfare between, you know, like, and I think that a lot of the scientists, by the way, who are good communicators do this. I mean, Lynn Body, for example, and her thing about white rot warfare zones. You know, this was her way to present it. And it's great. And it gets a lot more people both interested and understanding it.
I'll certainly never look at spalted maple without thinking about war zones.
Right. And she's absolutely right. They are war zones. And by the way, I wish that I had there's another slide that I could have showed, which is that this sort of sister group of mushrooms related to the oysters also had chemical weapons to kill nematodes. But at some point in their evolution, they went to physical weapons. And one scientist published a paper in which he showed how these microscopic weapons that this group of mushrooms has developed is exactly parallel to medieval weapons of warfare.
So that shows connection too. You know, you see similar techniques evolving in different parts of the human world in different ancient times, and perhaps, you know, as above, so below. And so thank you so much. I'm going to end the recording, Susan, but then I'm going to make sure you see all of the thank yous and appreciations that are coming through in the meeting chat. Thank you, everybody.